In generally, we have a mail-in sets. Now we're also using the mail-in also for size exclusion coupled SAXS. And you get all this information on our homepage that you can allocate your time. I will show you in details a little bit how to do it. You receive you registered a SIBYLS account, and you need to also register by LS, but it's not helping us, but LS to count the users. You book your slot. We also ask our users the same for the high throughput and also for the topics SEC-SAXS to apply for a rapid proposal. What is a formality, but again, it's important for LS to count the users and also give some plus points for the Beamline. But we're not requesting you doing every time we send the sample, please 3 times send the samples, 4 times you just apply for the proposal. You upload your information, you ship the sample or hand it to us. And this during the COVID time was much more difficult to hand on samples, but we've still been able to go out the gate and ask you to meet us out of this LBML gate if you are local. We collect the data, analyze the data, you receive the package, and you publish and you cite the SIBYLS Beamline. This was already shown, and I'm going to go through this. Our page, here you can see BL31LSLBL.go on HD-SAXS. You have all the information about the process of getting the data, sending the samples under this tabs here on top. Very important when you go book the slot. Apologize. You have options between SEC-SAXS and high throughput SAXS. You may click on it, you book your time. And now it is very important to read this. By clicking, you're accepting and you agree. And this is very important. You read the instruction. And I also refer to the frequent users because we still have sending emails back and forth, like you forget upload the buffer conditions, or you forget to tell us which column do you want to use for SEC-SAXS. So this information is very important to how do you gonna go to this introductions to the information and introduction. And you submit the spreadsheet. And the spreadsheet is not just bother you with some informations, but it's very important, especially the buffer, just in case the buffer is not delivered. We have capability to prepare the 10x buffer 50 mils for you, but we rather receive the buffers from you. You choose the column. This is very important. We have 3 columns, 803, 802, and 804 SHORT-X, and all the information, what kind of size separation you can expect for these columns are in introduction, in instruction. And then we have another important slot, the sample names and sample volume. That's the minimum what you need to put in your sample spreadsheet. We allow 7 samples per user. And the concentration is again the question, what is the size? And Greg wasn't merely mentioned it, I was hoping. But for size exclusion SAXS, you can imagine you have a separation in a column. So you need to have a little bit higher concentration than for the batch mode, for the high throughput SAXS. So I would say when you have a larger molecule, like about 100 kilodalton protein, what is, or 100 kilodalton macromolecule, you can expect with 3 mg per ml, with the 4 or 5 dilutions, you will collect the data from 0.5, 0.8 mg per ml. It's still possible to get a decent data for such a low concentration on size exclusion SAXS, but you need to have a larger molecule. If you have a smaller molecule like 20 kilodalton, you can't get any signal from the SAXS when you send us 2 mg per ml. That's dilution will just basically not give you a signal decent to really get any information. Expected molecular weights is just for our checkpoints. So all those others additional comments are not necessary, but it's helped us sometimes troubleshoot when we see that something is wrong. And then we see that it's not a problem on our side, but it's really the sample. So those information also go to our spreadsheet that we can start the robotics. So how this works, before that, the last year we have, even during the COVID time, we have about 184 size exclusion coupled SAXS shift, and these are the users, just individual labs. As you can see, the SHE lab from UC Davis, now in UCB, is the top runner. We have our local users as well, but also all over the US and also abroad. We use the Beamline in the size exclusion couple mode. So what's the size exclusion coupled SAXS just mean? In generally, we have a column that separate based on a size. We inject between 50 to 100 microliter sample. We have a robotic system to take it straight from the plate, but using us the Eppendorfs, and we just transport your sample after spinning to the plate, and the robotics start the multiple users overnight. Hopefully without any troubles, we collect multiple users over the days of size exclusion shifts. X-rays hit us relatively clean samples. Already Greg showed you how it works, an actual zoom in at the sample cell, and then we collect the data, 2D images, and we for each frame, for each time, we collect one scattering profile. And that's very important because you can separate different species, different oligomerization states, so you obtain a signal for multiple states of your sample. What we also add afterwards, and what other beamlines do as well, they're adding multi-angle light scattering. Why it is important, you will see in a second. This is just example. You immediately can compare those UV or light scattering signal, and you see the same what you can see at your home with your size exclusion. What is the separation? What is the shift after interacting with the components? And additionally, you had information about the molecular weight across the peak. In reality, as I mentioned, we have multiple users. We can run 8 users for one single run. That's mean that without attending, it's a kind of pseudo high throughput size exclusion, that we can start the data collection for 8 users when our system can exchange and equilibrate different columns without that we doing anything. But that's required a kind of a large amount of pre-programming. That's why we need all the information, which buffer and what column do you need. And we collect SAXS and UV moles and reflectivity index. And all this information you can extract from this multiple signals. It's just the SAXS, but also the form factor that's basically defining the solution scattering pattern. And from the moles, you have a molecular weight across the peak, size exclusion peak, and additionally hydrodynamic radius. So here, what you get back. In generally, we are running now a system that we are collecting data, analyzing SAXS and moles for you. You get a small report for SAXS that looks like this, and it's SAXS report PDF. For each sample, you also get a PDF file for the moles UV reflectivity index signal with analyzing the molecular weight across the peak. And that PDF file have also some values for you. What is the molecular weight of your main peak or additional peaks as well, like in this case. Some users ask me, oh, you have only 2 samples informations for SAXS, but you send 7 samples. You still have 7 PDF files for the moles. And obviously, if you see something like this, oops, then you kind of cannot expect anything from this sample. You see there is just aggregations, and the UV signal for actual sample is just not existent. Versus this, even that is pretty small amount of aggregation being detected, but you can separate aggregation to some kind of front peak here, and then you have the main peak on a side that you can analyze, and you can get the decent signals. You can see this dark blue is coming from this peak. So that's why you check the moles and check those PDF reports before you're asking the questions, oh, where is my SAXS data for this sample? This is exactly shown here. There is no signal that we can detect. Why it could be, we have also cleaning columns before the main column, and that may separate all the aggregations, and no signal can be detected from the rest of the sample. Okay. Why we, yeah, there's a question?
Yeah, quick question, because I've seen this before on my samples. Could you go to the previous slide?
Yeah, absolutely.
You would expect that if you have aggregation, it would go to your void, which is say 8 mils or whatever. But in this case, the green sort of is coming right at the beginning of the peak. I mean, or at the beginning of the run.
Oh, this one? Okay, so this is relative scale. So you don't, you need to look at actually the UV signal. And so the intensity of UV signal is 0.0 something. So this is basically zero baseline. Everything what you see here is zero baseline. Yeah.
Okay, sorry.
Yeah, it's just very, very zoomed in because this is relative scale. Yeah, that's a good point. Sorry. Okay. Why we doing the separation? Back then in 2007, we had a review in quaternary BioSAXS reviews, and I was trying to there explain that when you look at the SAXS data, this is shown as a PRL function. It's a pair distribution function from the SAXS curves, and you kind of see that when you have a mixture of oligomers and monomers, that's the blue curve. And when you expecting some movement in your in the secondary domain, and you kind of model this scattering profiles from these extended conformers, that's the red curve. And they are very similar to each other, and they are similar to the black curve. And the black curve is indeed your experimental data. So you had a hard time to distinguish if it's 20% of impurity of some non-specific aggregations, or it is really dynamic structure. And that's why I recommend do the size exclusion coupled SAXS, not only for protein complexes, but also when you're starting to thinking about some structural modeling, using ab initio shaping as well, but rigid body modeling too. So in this case, just another example, this is another protein. You barely see aggregation in the UV, so you think your sample is super clean. But when we're doing in the batch mode, there's no SEC, it's the red curve. But this is only zoomed in the small angles. Where we separate this on a size exclusion coupled SAXS, that's the black curve. And these are the Guinea region. And we may not go in the details right now, what the Guinea is, but we're gonna talk about it probably next day. But basically this indicate aggregation state of the sample, that's this initial part of the curve. And then we calculate this, as I mentioned, pair distribution function from the scattering profiles. And those data are extremely convincing there is something big and huge in case you're not using size exclusion coupled SAXS, rather just the batch mode. And obviously, if you just model that without knowing all these details, you're gonna get this kind of shape, and you try to interpret the scattering, this envelope as an opening of domain or any kind of conformational change of your ligase, what is this guy, then you kind of need to interpret your SAXS data, absolutely. And so that's, in this case, you don't see it on your labs size exclusion couple, size exclusion that, oh, there is super clean peak, one peak, but there is a little amount of aggregation can really screw up your signal and kind of distract this as a whale between the small fish. Okay, so I would say the application help us to really understand what we're doing here. This chapter. This is my favorite example, and it was from Mark Glover group, and it's again a protein DNA coupled protein DNA complex. But this is PP1, but it's also a hot protein target. But don't want to go into the biology. It's lots of biotech companies are focusing on PP1 these days, and Mark Glover solved the structure and his group. And this was the crystal structure, and they see some conformational flexibility between ASPP and PP1, but have this huge linker sticking outside. Interestingly, he found interface between the SPP1 and PP1. And so he predicted some flexibility and some motion. But when you look at here, this is the UV signal, and overlay the SAXS signal is shown in the blue, but it's not the SAXS signal indeed, but it's the radial duration for each frame that we collect the beamline. And so you see it's declining across the peak. However, when you look at the multi-angle light scattering signal, what is the molecular weight, is flat, at least in the region where the RG is declining. And so that was all the time the question, so how is it possible that we have a different size based on the solution scattering, but the same size based on the molecular multi-angle light scattering? And one of the reasons is this, that the X-ray gives us the conformational state versus molecular weight just define the molecular weight because of the resolution of the technique. And that's the beauty of the SAXS coupled with the multi-angle light scattering. Then we came to the modeling and we tried to fit the data, the SAXS data not showing here, but they come up with the different conformers, open and closed, and where the PP1 is holding basically ASPP in different states where you have a transient or multiple interaction between ASPP and PP1. Hopefully I said it right, but that's allowed us basically partially separate those intermediate states because you have a multiple interaction that can hold the ASPP in a place, not in a place, in a very dynamic state. And this modeling was done with the Bilbo MD. It's one of our tools that Greg mentioned in his introduction, and it's also accessible with simple VAP application on our page. And so that's how we analyze this kind of data, that when the crystal structure do not fit the data with a really bad chi-square versus chi-square 1.0, where it's mostly optimal or perfect when you implement this kind of extended conformers and a multiple of them.
Michal, just to let you know, there are questions in the chat.
Okay.
And we can either read them or we can have people ask them to ask the question.
I would ask somebody can just unmute himself and say hello and say what question is. I would like to hear the voices as well. I don't like to hear my voice. Who's going first?
I think Grace had a question.
Grace,
are you there?
Yes, I am, and he kind of touched on it in the subsequent slides.
Oh, I will. Yeah, Grace, I see your questions. I will continue more about it in the 3 minutes. Yeah, okay. Perfect.
Thank you.
Yeah. Rob was asking the question. Oh, Rob answered the question. Perfect. Thank you, Rob. I would like to hear your voice too. Okay, great. Matthew, have you a couple other separation technique? Oh, okay, that's a good question. We are working on it. We're thinking about different columns, not only size exclusion coupled SAXS, but key columns or any exchange are doable. It's more difficult because you have a salt gradient in this kind of system. But there is an application, I think, in Grenoble. They don't use it so often. Again, the subtracting the buffer from the peak of interest is not trivial. But yes, I was thinking also for DNA protein complexes, sometimes you can use the DNA columns as specific design for separating the free DNA for protein complex. That may be also useful. But yes, that's our future. So far, we have so many users with the size exclusion, and I'll be happy with that. I need to watch out my time. It's still okay. I will try to give you some real application, not only talk about what we did over the past time. SAXS during pandemic. I was really proud on Greg and our political influence at the LS that we've been able to open it in already, I think, in beginning of April. And we, not me, but Daniel was able to work at the Beamline and collect the data, not only for the COVID project, but we said it's for the COVID only, but we mostly collect a COVID related projects. And this is one of the examples where we collect the SARS-CoV-2 replication complex with different RNAs and different components. As you may know, it's not only about the spike, but it's also about how the SARS-CoV-2 is replicated. And that's using polymerase, it's number 12, NSP12, non-structured protein 12, and we have lots of cofactor 8 and 7. And so we find out that, for example, just only look at the, this is the, not UVC, this is the light scattering, but you can consider it's a UV signal. And we can just immediately see the 12 by itself. It's not really stable, you have multiple peaks here, but when you have a number 8, on it, it's make complex. And what was surprising to us when we add RNA, there's no complexation happens. So when you have the number 8 and 12 together, RNA, it's not binding this 8 and 12 together. But this completely changed when you give to the polymerase what is number 12, this other component, what is called NSP7. It's immediately make this shift, and very super strong signal show us it's a very stable complex. So we have 7, 8, 12, and it's bind the single-stranded RNA as well, the double-stranded RNA. Just looking at these few samples tell us what's happening with the system, how it's behaved. And we did it with DNA, it's not binding the DNA, etc. So we published this story with also analyzing exactly the SAXS data. We have information from cryo-EM, and so we had opportunity to match this data from cryo-EM, the atomic structure, directly to the SAXS. And so you can see the models are in the dotted line versus no, this is not the model. Sorry, we're just comparing here different complexes, but in generally, we calculate and find out that only when this tiny small NSP7 is present, that RNA can be complex with this replication transcription complex. There is a multiple information that we additionally add to that, but it was just straightforward technique that we applied, the side-exclusion coupled SAXS together with the molecular weights, shift of the peak, and simple modeling. Luckily, the cryo-EM came like really a couple of weeks before we have the access to this data. So this good combination tell us and multiple stories how the machinery works, and which DNA, why DNA is not binding, where supposed to RNA probably bind, and confirm most of the cryo-EM structures. That was one of the happy story. Another happy story that I worked with Curtis was look at the antibodies against the SARS-CoV-2, and then that great project again, because we have this show the high throughput, we can run multiple samples. So we got access to some funding. We bought a bunch of antibodies against nucleocapsid. Nucleocapsid is the protein in the virus that wrap the RNA and form basically the core of the capsid, of the viral capsid. The question was, we have only small, tiny domain from the nucleocapsid protein. And the question was, can we use this nucleocapsid protein for rapid test? So how we can improve the rapid test with coupling and choosing the right antibodies against the nucleocapsid and terminal domain that is immediately released in the rapid test after you put your swab in a test tube, etc., etc. So that we test and we immediately see which antibodies didn't bind versus the antibodies that bind. We can see it in the SAXS curves as well, so it can be used as a screening technique. Just checking the RGs could be enough, where binders and non-binders can be distinguished. But what you can also do, because we have general structure of antibody, that's how it's like. So we can calculate scattering profiles from these antibodies and match our data. This is just antibodies itself. And what we find out that after matching this atomistic model to the SAXS data, we need at least 2 conformers to match the scattering profiles. What tell us that we know that before, but just want to repeat it for those that are not expert in antibodies. We have this T and Y conformers, so I would say open and closed conformers. So we also try to calculate some histograms. So all these tools are available. And what we find out, there are antibodies, they are very rigid or they're very flexible. And now we had the questions how we can couple those antibodies to improve detection signal for detecting the nucleocapsid. What is the main target for rapid test? Currently, the COVID-19 rapid tests are used and they are available as well recently. Back in April, there was nothing available. And so we find out when you're using a flexible antibody that are able to make this kind of bridging, what is a kind of funny story. So we call it the sandwich type of bridging, because you're using in ELISA or in rapid test, you're using this kind of type of bridging of antigen and detecting it with the couples of antibodies, with coupling it. And so when you have this kind of sandwich type interaction, you cannot increase the signal versus when you have a very rigid antibodies, they form more linear conformers because they cannot adopt the sandwich-like conformations. And that pushes us to detecting lower concentration of nucleocapsid by using rigid antibodies. So it's not only the epitope recognition, but also general flexibility and dynamicity of antibodies what matter. And also, I think Rob is here. So we started using this SHOTX columns back when he was still in the lab because he thought it's a good try and it's really one of our front runner. And company asked us to use this publication as an advertisement for their columns, KW 800 or 400, and put it in the catalog page. We got some columns for free. So win-win on the both sides. So you guys, if you're using SIBYLS in the future, you're gonna use the free column from our main company that we're using for separation. I would like to speed up a little bit and go to the practical more, because tomorrow we will have a practical. But before we go there, I will choose one of the slides. Okay, so size exclusion SAXS and couple max we can use under anaerobic conditions. It's not easiest, but we still are able to do it. And that was very important, a project with one group at the UC Berkeley, and Daniel was involved with Horst to work out the system. Required exactly, what did I say, under anaerobic conditions, not only the buffer, but also the sampling to be prepared under anaerobic conditions. And we tried to figure it out, the solution state of nitric oxide receptors that we had cryo informations, but they could not see exactly the active state. So question was, what was the active state? And so using our UV, we can measure also different wavelengths. And with the 432 nanometer, you can detect if the active state, if the active site is occupied with NO or is not. So you can see the shift there. That was also important for using our pipeline, not only just collecting SAXS data, but also if the protein is not oxidized, if it's still in anaerobic conditions, et cetera, and how much NO is present in an active site. And obviously we have some kind of SAXS signal, we have some moles, and all is same in NO active and NO inactive state. And we just using the cryo-EM information, we add some linkers and come up with some idea how the activation by NO, you will see that the whole structure of nitric oxide receptor soluble now cyclase just open in a specific way. And this is again matching and implementing the cryo-EM together with the solution state. I don't have actually the SAXS fit for this kind of modeling here, but you can see that activation in our anaerobic size exclusion coupled SAXS can be visible in the actual SAXS data with the difference in the scattering profiles. There was a question about disordered. So this is one, again, it's very difficult to distinguish aggregation from intrinsically disordered protein. XOCC1 is one of those human proteins that work with the it's not base extinction repair, but single-stranded repair. For those people that are not expert in DNA repair, it's basically DNA repair pathway, coupled bunch of ligases, and it's kind of scaffold protein that hold those pieces together for continuation of the repair of the single-strand break. And XOCC1 is very long one, but have only few domains. So all of them being predicted as a disordered. And indeed, when you look at our SAXS data, here, that's the SAXS data, and we look at sometimes not as a PRR function, but we can look at as a Kratky plot. And hopefully somebody else after me going to talk about all this PRR function and Kratky plot. But when the peak of the Kratky plot is shifted from this line, what is the square root of 3, and I don't want to go in detail why. I think Rob's going to be spending hours about it, why this square root of 3. But basically, by looking at the peak position relative to this line, I can tell you how much disordered you have. And indeed, this is extremely disordered protein. Not only is shifted, but is not like belt-like shape. Here on top, you see the usual plot that I like to show all the time. It's the UV signal, and this is a SAXS signal. On top, you see the molecular weight across the peak and the radius duration. Again, you have a huge difference between peak position and the tail. So there's happening totally opposite story to PP1 that I showed before, but XOCC1 most likely have a different oligomerization state across the peak. And again, you have this information right here. Therefore, I recommend to tell everybody, before you analyze the SAXS data, look at our PDF reports for the moles. And it's make also Daniel really happy, because Daniel works hard to really help you with those moles data and give you this report. And that's indeed, it's happening here. It's just kind of this pink one is right here. The full length XOCC1 is a mixture of monomer dimers. And again, to match this data, and again, I'm matching here the PRR functions, where you see the experimental data in a color, that's the pink one, and the black one where you barely can see, but is overlaid, because those are atomistic models that are shown here. So you need to really think about it as not only as a mixture of monomer dimers, but also extremely unfolded regions. They're moving those N-termini central domains and C-terminal domains in very dynamic fashion. It's a similar way when you cut one of the domains, basically, or still stay monomer dimer mixture with very unfolded regions. So that, I think that how we can recognize this unfolding through the Kratky plot, and I tried to mention that not only Kratky plot, but also when you're expecting what is something about 50 kilodalton, and you see a Dmax of 250 angstrom, and you know your data is aggregation free because you've done size exclusion coupled SAXS, then you're really asking the question why my 50 kilodalton protein is 250 angstrom long. That's coming from this PRR function, and you see that when the PRR functions go to the 0, and I will just finish my thought and I will respond to the question. The PRR function 250 angstrom tell you that that's probably elongated, and indeed is unfolded, partially unfolded, as you see in Kratky plot. So combination of those two information between POR and Kratky plot can tell you, and indeed, then you have also the moles here that tell you, oh, you have monomer dimer mixture. Oh, it's a very elongated. Oh, it's a disorder. Bang, you're done. That was exactly what you're looking for, and not going to alpha fold and believe that what the alpha fold structure is, that's the only structure. Anyway, that was just a joke on the side. But the question was, what are the molecular weight size limits for size exclusion SAXS? We have some very large nanoparticles, ELPs, NLPs, perfect, that we are working with, interesting using technology. Yeah, so depending on if you have any experience with the size exclusion in your lab or maybe in your company, because I think we have some company attendees today here too. Pardon? Yeah.
That's my question.
Okay, Nicole.
Hi, so I'm actually at the NIH. So we were at Washington University and we've used your guys' system for a long time. We've transitioned now making nanoparticles and VLPs for vaccine development.
Yeah.
Yeah, and so we've sent some through with your SEC-SAXS, but the molecular weight cutoffs for your columns that you have, some of our nanoparticles are larger than that cutoff. And so I was just wondering if you had another way to do it.
Yeah, are you talking about lipid nanoparticles?
No, these are all protein-based, but they're multi-component.
You talk about some kind of apoferritine cages?
Yeah. So
well, I would,
so we have
I will show example of apoferritine because there was one user that was explicitly asking how should we analyze this data.
Yep, that was me.
Yeah, I was you. I will show that, and I think the separation is perfect because your size from the molecular weight is relatively small.
Yeah. So we have some. Yeah. We have some that are based off of like E2P and lumazine synthase that are much bigger, and so those are 60 MERS. Plus we have the antigen put on them. And some of them we've done things like the large spike proteins, and then that really pushes things very, very big.
We have also, and Somi are going to talk about it tomorrow. I hope she's going to show some failure and success as well. The spike itself is a little bit tricky. You have lots of glycosylation, and separation could be tricky because it aggregates when you're putting some nanobodies on it. It's just not as simple to say, I believe not aggregations, but make some kind of cross-linking, even that you're expecting one nanobody, et cetera, et cetera. For your apoferritine, I will show the example. It's not the problem the size. It's relatively the 804 column that we're using currently have 1.5 megadalton, and I think Rob can comment on the details about that. 1.5 megadalton is the size, the maximum, but we allow the users to send us their own frame rate column too. Okay. Especially when we talk about NIH project. I don't want to take the size, but I think during COVID and what we did with the SAXS, relatively simple technique, it was very useful for companies as well to apply this technique for screening, developing, checking the stability, etc., 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 etc. So yes, you are okay to send us your Super 6, and we will try our best. If the 1.5 megadalton is, it's
Michal?
not, yeah.
Sorry, Alim has some suggestions. Alim?
Okay, you need something other than SEC for separation. That's right. Yeah, Alim.
I do have a comment on this.
Yeah, go. Thank you.
So there's two things to think about here when it comes to SAXS and the size of things. One is for a beamline like SIBYLS, it has a fixed camera length. They have a maximum size that they could effectively measure, right? So you want to make sure that you're within the range of the camera. Otherwise, you need to find a facility that could change the camera length, right? So you have to ask Greg about, I think, whatever that is right now.
I would comment on that as well. So since Rob, you've been here, we did lots of upgrades. So even that we have a fixed camera, we extended the length. So the maximum size is about, I would say, 500 angstrom, 50 nanometers. So, so 50 nanometers apoferritine plus with antibodies plus with anything, it will be okay.
Okay. And then the other thing for separation of large things is, there's these standard columns that people have, the SuperDex, Superos, the Shodex KW800 series. So usually what we're seeing is like people are pushing the limits of that because they want to go to larger things, right? And these columns don't necessarily support that. But what we have seen is if you connect two of them together, so two 800 series columns or two SuperDex columns, you increase the separation resolution pretty significantly for large things. The caveat to that is that the run is going to take about 3 to 4 times longer because the back pressure goes up and you have to run at a lower flow rate. It's completely worth doing, but you have to convince the Beamline scientists to do this, for one thing, or you have to apply for time. You have to apply for your own user time and do that with the Beamline scientists, right?
Yeah.
But it does work, and you can get some pretty amazing results.
Yeah, well, we had a success with Super 6. That's, I would say, try that first. That's
Yeah,
go to
we've been using that one, and then we also have another column that we've been using. I have to check with my tech who's been doing it, but it's more of an HPLC column that we've cobbled onto our FPLCs that we've been using.
Yeah, if I will have a time, I will show you upper ferritin with the, I think it was a 20 kilodalton protein on top of it. So,
Yeah.
and you will see it's worked pretty much perfectly.
Yeah, that
that's
was
not
my
a-
data. So
Yeah, okay, I have that.
thank you.
Yeah, thank you, Rob. That's a useful info. I never tried to have 2 columns back to back. I think that's worth to try it. We have also users, they're using it. Sorry, I'm living in the mosquito crowd here. So we have also users that are really exploring size exclusion SAXS in every sample is valuable, even that you think this is not true. The complexation with different components, I already mentioned that. This is just one of our users that explicitly using size exclusion SAXS. And GP5, those are, anyway, some kind of polymerases. Oh.
I could jump in. It's my data.
Tell me,
this
tell me
is the
what
bacteria. This is a bacteriophage T7 DNA replication system.
T7 replicate.
Yeah,
Yes,
where we were trying to look at complex assembly, and we got this data, and then shortly thereafter the cranium structure came out and agreed well with it.
Perfect, thank you. I would just want to throw you this slide that when you compare those data, what we're sending to you, it's very important to compare SAXS, not only just as a curve, because it's not going to tell you so much, but it's a PRR function. You see the complexation. You can confirm that with the UV signal or even the molecular weight, what you see right here, illusions. You can overlay that as I show in the slides before. So all that information can be used not only as a structure information, but as an assay. And I will try to show it on those apoferritin samples you have at time. And I'm not sure how long I'm supposed to talk. It's just an application. Yes, here we go.
You have about 5 more minutes, Michal.
Perfect. That's great. And I really appreciate that there are questions that people asking, interrupting. I don't like to talk about amazing science, but this is a workshop. So I will really give you more hints what to do and how to do, but I will show you how simple it is. So again, we have Rob Rambo. He was the main, he started to develop this tool scatter here at the SIBYLS Beamline. Now he have definitely more time and he's putting more tools together. So he's making better and better every day. Hopefully the version 5 will be cleaner, better, and nicer. More colors. We want to see more colors. Anyway, that was just an afternoon talk because I'm in 9 hours ahead for front of you. I need to find this. See, I think this one. Anyway, so simply what you get the package from us, you get also this SEC file. That's basically, you don't need to upload all those dot files that you have also in subdirectory, but you can grab that SEC and put in here, and you will see this is your peak. And so also you will see what background I selected. I selected the background here. You can select the whole region, but I was kind of lazy. You can also select, if you have some problems with the buffer subtraction before and after the peak, you can adjust your buffer subtraction by set the buffer, So you adjust the buffer. This is just very brief. We're gonna go in details tomorrow, but because that was a nice discussion about vaccine development. And when you select the speed, you will see that it's relatively 56 kilodalton across here. It's relatively stable. There is some capillary falling behind, so we don't want to take this region. This signal is kind of weird because there's no signal or not enough. So that's what I will select. By selecting the region here, you can say this is my folia 1. And I say merge the data. And when you look at the analysis stuff, you'll see your data. Okay. And we're gonna click off the R2RG. Anyway, this is your data. It's a beautiful data for apoferritin. And if you want to compare this apoferritin with the data that somebody already previously published, or you can go to SIBYLS, and that's a deposit for a different SAXS data. We're still using it for our stuff too. Let's see if there's apoferritin. Oh yeah, so you see there are some apoferritin even collected on the size exclusion coupled SAXS. Obviously, this apoferritin is not in size exclusion coupled SAXS and have a different RG than the size exclusion coupled SAXS collected apoferritin. And you can upload this data compared with your data sets. So just be sure that what we're delivering, it's about 400, 500 kilodalton. Perfect. So this is your data, this is your raw function. So you can get this information from here and you can compare what you got. Let's just compare your apoferritin with most likely some small antibody on it. I'm just guessing 20 kilodalton, that's it. But I was just guessing. So you clear the samples and take your sample with, and you already see the signal previously was here about the frame 420, 450. And you should look at the PDF report for the most data to confirm the molecular weight. I think in this case, I couldn't find the PDF file for the molecular weight. But let's just do only with the SAXS. Again, something wrong here because the RG is so huge here. So ignore those values here. But obviously, you have a different, based on a similarity map, and Rob's gonna mention that probably here, based on a similarity map across the peak, you see those frames are very different across the peak, especially in the region of the main peak. So you have a different scattering profiles, but most likely is the partial complexation of your apoferritin with your recognition or protein, not antibody, some protein that is functioning as antigen. So that is already seen here. And you can also select this region and say, this is my curve 1, and you merge that. And you can also compare with this region and call it as a curve 2. What you do here, and indeed, when you compare the black one versus blue one, they are a different size. You can see it from the RG. Not really. So they are same. So I was wrong. Or I think that was just the buffer construction. So ignore the 2, ignore the 1, or ignore the number 2, and just look at the difference between. And you obviously see the complexation here. You have a different RG. You can calculate the RG by clicking here, and I think Rob's gonna just talk about that in details.
Michal, sorry, I just checking the time. A couple minutes, yeah. Thanks.
Okay, so I will just on our show respond to fast questions today from the user. And so you kind of can confirm that your apoferritin is 54 kilo RG, what is exactly what was deposit in a different from different group versus RG 78. and obviously you have the complexation. To model this kind of system is a little bit more tricky, and we're going to talk about it tomorrow, but it's a very fast way to see in a real space how those pair distribution functions are different. You just, oops, it's not going to work now, but you need to trust me.
We have a question in the chat.
Okay. Just finishing it up. And I'm gonna go there in a minute. Okay, so what I want to show to a user, what I will do as a main point of interest. First of all, we show that across the peak, it seems it's very stable. The scattering profiles are very similar across the peak. That's indicated probably a stable compensation and not a partial compensation. And also this PRA function, what is the fair distribution function, that's the main information what you probably can get. And you see the huge difference between apophyretin itself and the complex apophyretin with DFLB or some kind of antigen. And so all this information is just the initial analysis. And if you want to model that, you need to go to more complex stories, but we're going to talk about it tomorrow. So this is very fast, how you look at the data and how you confirm, you get a compensation. And you can calculate molecular weight from this to confirm that, or look at the most data that you have. I'm going to look at the question now. Can SIBYLS distinguish small size of aggregations like the peptide dimers and trimers? Of course, that's a good question. I just bring, you see this peak, for example, I just bring something here. That's a small aggregation of NSP12. And if you not separate this from this main peak, you will not able get this beautiful data and match your cryo-EM structure with your SAXS and make this conclusion that without NSP7, you will not, you have not a DNA or RNA complexation. The same, for example, this is a complex, in this case, NSP7, 8, and 12, without RNA is very transient. Again, you see the small shoulder here and some another peak here. Let's tell you that without RNA, it's a very transient behavior, and you have not only from the molecular weight indication, but also from the UV signal, or in this case, the light scattering signal that is very transient. And you can separate, in this case, a small differences between NSP 12, 7, 8 versus NSP 12 and 8. So small details could be this, and depending on the column that you use as well. So you need to adjust the column usage to the size that you have. Oligomers is the main, and everything. I mean, this is another example when you have hexamer versus octamer. I don't want to go in details here. Everywhere where we look at some significant, you see, this is a project that started, this is ASPP, PP1, it is small unspecific dimers or whatever it is, maybe some coupling of two PP1s or two ASPP with one PP1. We need to separate this signal before we can do all this beautiful modeling and come up with the models. One more questions?
So I have a comment too.
Yeah.
So the problem with really small peptides, so we measure peptides a lot here at Diamond, is the smaller you are, the larger your Guénier region is. And so what ends up happening is that, you know, if you measure something like sucrose or the size of sucrose for SAXS, you just measure a really long line, right? So as you have to figure out roughly, you know, if it's something on the order of like 8 amino acids and it's a monomer, you know, 8 residue peptide, you're really just going to measure a Guénier region, right?
Uh-huh.
And then what you're really thinking about now is if it's going to aggregate, can I distinguish the dimer from a trimer, et cetera? And that really depends on how the RG is going to change, right? For really small things. So the best thing you could probably do is just try and model it first. Greg has always said this is a really easy thing to do, is just build a simple model, use Foxes, model out what the beamline is, and see if you can measure something that's going to be meaningful. And then, put two together, make a dimer, and see if you can measure that change using Foxes, or it shows a big change. And that's going to give you some sense that the beamline, the SAXS measurement is going to work for you.
Yeah, that's a good point. I didn't have a time to go to the peptide, but yes, separating monomer to dimer peptide, it's not that tricky. But I think when you have a high concentration ROP and when you're using the right column, definitely not a shortcut system, then you are able to separate the peptide and collect at least the thread line, the RG may indicate that you have a different species. I think that's a good point. We have a success even with small compound, because compounds, some of the small drugs can form aggregations or trimers or tetramers, and we are able to separate from the single species and see stuff like this. It's really need to be tuned up, and you know your system better than we do. So all these changes in a column is, as Rob said, you need to be nice to Beamline scientists. How small issues the column can effectively solve their gates? Something. Oh yeah, that was the response. Okay, I think the rest gonna continue tomorrow or over the today discussion.